Today we'll be learning about lipids and lipid bilayers. Here are the learning outcomes for the day. We're going to recognize the four main classes of lipid. We will be able to look at the structure of each of these lipids and translate that structure into line stick, and then understand kind of the overall shape and translate that to its van der Waals radius. We're going to look at the different types of movement within the lipid bilayer and be able to understand how fluid that membrane is. We will compare and contrast the three types of membrane-associated protein and kind of understand each of them. And then finally, we will see another example of a hydropathy plot and how to use it in this case for a protein going through the lipid bilayer. Today's Meet a Scientist moment is Professor Bill Clemens Jr. He is a professor of biochemistry at Caltech, and he is a Black scientist. And he does some pretty interesting research relevant to today's topic in that he studies the structure and synthesis of membrane proteins. This is particularly challenging work to do with proteins that are within the lipid bilayer because they are really stuck into that lipid bilayer. So understanding how they're synthesized and understanding their shape and structure becomes very challenging. So let's talk about this next class of biomolecule, lipids. This is going to be the last one we discuss. We have talked about protein, we have talked about carbohydrates, we have talked about nucleic acid, and now we're going to talk about lipids. Lipids are not polymeric, similar to carbohydrates. They are polymeric. Nucleic acid, that's a polymer. Proteins are a polymer of amino acid, right? Lipids, not polymeric. But their biggest function that we'll talk about today happens when they aggregate with one another. They are incredibly important, especially to the function of our cells, hence the lipid bilayer named after lipids. But they're also important for energy storage, so if you take biochemistry 2, you'll probably learn more about this. And they are important for signaling, so if you've taken any cell and molecular class, you might learn a little bit more about that then. There are four main types of lipid that I want to cover today, and I want us to have some type of recognition, just visual recognition for these lipids. There are fatty acids, triacylglycerol or triglycerides, phospholipids, and steroids. So let's start with the first. Fatty acids. So this is the simplest form of lipid. This is kind of like the OG lipid. It is simply a long hydrocarbon chain with that carboxyl functional group at the top. Now we call this type of lipid amphiphilic, or synonymous amphipathic, because it has both a long nonpolar region and a polar region. So things that are amphiphilic are both polar and nonpolar. The most common lengths of these type of lipids have 18 or 16 carbons, and it's, at least in humans, usually an even number because we build our lipids 2 carbons at a time. But I do want to make a quick note that if we're looking at these lipids, we want to be thinking, okay, to understand their function, we need to know what they look like at physiological pH, right? Same story with amino acids, where the N-terminus is generally protonated and the C-terminus is deprotonated. You might assume, like the C-terminus, that the carboxyl functional group is deprotonated at physiological pH, giving us a carboxylate. If you assumed that, your assumption would be right. So, for example, let's just draw out this first lipid as it would appear at physiological pH. Let's start with that carboxylate functional group, deprotonated, negatively charged. Now we're going to have 18 carbons, so hopefully I can count to 18. The first carbon being counted, as you can see on the right-hand side of my screen here, is the carboxyl carbon. So I'm going to start with that being 1, and I'll count up to 18. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 carbons. So this is more representative of what a fatty acid would look like at physiological pH. Now, as you might already know, we can classify fatty acids according to how many double bonds they have, if any. On the previous slide, you already saw that I was showing you no double bonds, one double bond, or more than one double bond. And so we can classify these fatty acids according to their saturation. Fatty acids that are fully saturated have no double bonds. They are fully saturated with hydrogen, right? They have maxed out their hydrogens. The lowest energy conformation of these lipids is extended, so that's why we would draw them, like on the previous slide, just as a straight chain of carbons. Now, since these are very linear, they pack together well, they can snuggle up right next to each other, and they lead to a higher melting point. So think butter, right? Now we can compare this to unsaturated or polyunsaturated fatty acids. So here we're talking about any fatty acid except for the one on the left. So one double bond or more than one double bond is unsaturated or polyunsaturated. These have double bonds almost always in the cis configuration. There should be a 30-degree bend in the chain. So let's draw what that means for us here, and I'll do my best because it's sometimes challenging to draw this realistically. So I'm going to start drawing this lipid and do my best to show you what I mean by a 30-degree bend. Okay, so let's say we have a double bond here. If this double bond is going to be cis, I need the next bond to come off in the same direction as the other alkyl chain, right? So let's draw this as best as we can. And then we just continue the chain. Okay, so what you can see is that I've created kind of this 30-degree bend in the chain. And this is what an unsaturated or polyunsaturated fatty acid would look like. The most common place to see the double bond, if there's one or if there's more than one, is at the C9 position, and hopefully I counted that out right when I drew it for you. Now, these types of lipids do not pack together super efficiently, right? They're bending off on all types of angles at different carbons. So they're going to kind of have less interactions with one another. This reduces the nonpolar to nonpolar interactions, the London dispersion interactions between these lipid tails. So this results in lipids that tend to be a liquid at room temperature. So think olive oil, avocado oil, right? There are two important classes of polyunsaturated fatty acid that you are probably familiar with, omega-3 and omega-6 fatty acids. Up next, we'll talk about the next three types of lipid.